So now we move to our short lightning talks. And the first one is given by Arnoud van Vliet, and he will talk also about AMR in markers in Campylobacter jejuni and C. coli.
Thanks, Cynthia. So I want to talk about antimicrobial resistance to build on what Desi was presenting. So this is a collaboration with North Carolina State University and people at the University of Surrey. So firstly, if we look at genome sequencing, we heard a lot about it for the last 2 days. It's now the gold standard for epidemiology, but also for AMR screening. And there are now more than 60,000 Campylobacter genomes available in GenBank and PubAmnesty. But there is a potential bias of people sequencing interesting isolates, for instance, ones with lots of AMR genes or special virulent strains. So they may not be an unbiased representation of what is out there. For that, you really need these unbiased surveillance programs where just everything is sequenced that is brought in. There's 2 countries that really have that. The UK has the FSA Oxfordshire Sentinel surveillance. They focus on clinical isolates, but of course, most of those clinical isolates will have come from sources like chickens, bovine, environment, etc. And the USA started Genome Tracker, which basically focuses on food safety, and NARMS, which focuses on AMR. And they really sequence everything from farm, shops, and clinical isolates. So it's a much better representation of what is out there and really large numbers. Now, we need to use the concept of antimicrobial stewardship, which is a set of coordinated strategies to improve the use of antimicrobial medications. And one of the goals is to reduce antibiotic resistance. And very simplified, the concept is that antibiotic resistance is expensive for bacteria. And so if you grow without antibiotic, the resistant bacteria should be outcompeted in the absence by the susceptible ones. But is this really true, and is this working? So the aim of the study was to check whether the reduction in antimicrobial uses in the UK, due to the EU guidelines in the early 2000s, and the USA a bit later, has it actually reduced AMR prevalence in both countries. So we had a total of over 40,000 jejuni and coli genomes. We had the metadata available for those, so the year, country, and the source. We used AMR Finder to predict antibiotic resistance. And so if there was a gene or a mutation that is predicted to give antimicrobial resistance, then it was counted. And we focused on 4 main classes of antibiotics relevant for Campylobacter. Very quickly, just to show the data set. So these are for 2015 to 2018, the number of Campylobacter coli sequences, in blue the UK, in red the US. You can see from 2015 the US surveillance taking off. Same for jejuni. You see the US surveillance taking off, whereas for the UK it stays more stable, although it goes up and down in jejuni. And here you see the sources for jejuni. You see why the UK one is very focused on clinical isolates, whereas it's a more distributed types of sources for the US. And here you see for the 4 main Campylobacter relevant antimicrobials, so the aminoglycosides, collection of 20 different genes, the macrolides, mostly 23S mutations and a few genes, the quinolones, mostly GyrA mutations and a couple of genes, and tetracycline. And what you see here is that the 2001 to 2014 combined has been included for comparison, is that macrolides, the US, is very clearly higher in C. coli and also in C. jejuni, although it's quite a bit lower, sorry, for the aminoglycosides. For the macrolides, you see the US going down and the UK going down as well, and it keeps low in jejuni. In quinolones, actually, the UK is higher than the US. And in tetracycline, the US is a bit higher, but you don't really see, except for the macrolides, any real reduction in the antimicrobial resistance prevalence. And it seems to be almost like it's a stable resistance. So macrolide resistance, luckily for C. jejuni, is very low. And although most of these are significantly different, the differences between the UK and the US are not particularly strong. We did look at source and MLST types, and it was mostly MLST types, like Desi shows for fluoroquinolone resistance. So to conclude, despite reduction in the antibiotic usage, there is no clear reduction of AMR in Campylobacter in both countries. So does that mean antimicrobial stewardship is failing and can only control expansion of antimicrobial resistance, but not lead to further reduction? And for the 4 classes, don't use tetracyclines, don't use aminoglycosides, especially not in C. coli. Quinolones are very much of concern because of ciprofloxacin being used in humans. Luckily, the only good news is macrolide resistance in C. jejuni is very low, and C. coli, it's more of a concern. So I hope I stayed within time. That's it.
Arnoud, thank you very much. Yep, you stayed within time. So please, any questions, I encourage you to put them on Discord and we will go through them at the end of the session with the speakers. Okay.
We shouldn't answer them in the chat yet.
For the lightning sessions, I would encourage answering them at the end orally so that everybody can hear them. So the next speaker is Dennis Linton. Dennis, if you'd like to share your screen.
Okay, thank you very much for the invitation to talk today about some of our work on metal exporters in Campylobacter jejuni. So yes, I'm going to talk about metal ion homeostasis in bacteria. And to maintain metal ion homeostasis is very important. And to do this, bacteria use metal uptake systems and they use metal export systems. And they're using these along with sequestration, intracellular sequestration, they can control the levels of an array of biologically important metals within the cell. And this is key to proper cell functioning. Now, the host during infection tries to disrupt this, and they do this by restricting the availability of metals in a process known as nutritional immunity, and iron is a very good example of that. And they also poison microbes with antimicrobial concentrations of metals such as zinc and copper in the so-called brass dagger model. Now, microbes, again, they respond to try and maintain metal homeostasis during infection. And one of those tools is to produce metal exporters. So I'm going to talk about this in Campylobacter jejuni. Now, metal exporters belong to a number of different structural families. And the one I'm going to talk about today is cation diffusion facilitator family, indicated here. These are dimeric, inner membrane located proteins with 6 transmembrane domains in the N-terminal region and a soluble cytoplasmic C-terminal domain, and they function to export metals as a proton antiporter. Campylobacter jejuni has 2 of these cation diffusion facilitator family proteins, and the first is CZCD, and we've been studying both of these. We have inactivated this gene and created a genetic complement, and what we've shown is that the mutant is hypersensitive to zinc compared to wild type and complement, and overaccumulates zinc. This gene is also regulated by zinc. So if we increase zinc, we see more CZCD protein, and this is at least in part due to this promoter here being activated. So when we make a fusion to GFP, we see increased levels when we add zinc. This is also regulated by a really interesting genetic element known as an RNA thermometer, which is located just upstream of CZCD. And what this means is that CZCD is produced more at elevated host body temperatures, as indicated here. And finally, this CZCD protein is regulated by iron. So this is unpublished data. Iron represses production of CZCD, and this operates through this region here through a mechanism that we really don't fully understand. So not through this promoter, as far as we can tell. So what you have is a protein that's produced in high zinc, low iron, and high body temperature, indicating a role in virulence. And when we look in the Galleria infection model, we can see that the mutant is attenuated in green here, compared to wild type and complemented strain. And intriguingly, the mutant is also more sensitive to hydrogen peroxide compared to wild type and complemented strains. So the second example of a CDF family member is a protein called CJ0948, and this is unpublished data. And we've inactivated this and again made a genetically complemented strain. So this is located in a 3-gene operon. And when we inactivate CJ0948, we now see not zinc sensitivity, but iron sensitivity. And we also see accumulation of iron in the cell in the mutant. So this indicates that CJ0948 is now an iron exporter. When we look at more virulent related phenotypes, again in Galeria, we see that 0948 is again attenuated. The mutant is attenuated in the larval injection model compared to the wild type. And it's more sensitive to hydrogen peroxide. And in very recent data, we've shown that when we use a competitive index experiment with wild type and mutant, the mutant is outcompeted by the wild type in a macrophage survival assay, indicating a role in CJ0948 in intramacrophage survival. So we are currently investigating the regulatory inputs into this gene cluster, which we think may well be interesting. So to summarize, the key messages are that Campylobacter jejuni has 2 of these CDF metal exporters. They have roles in zinc and iron export, respectively. There's complex regulation for CZCD, and the data suggests roles in infection during infection. So we propose that host-induced metal intoxication in C. jejuni infection requires further investigation. To finish, I'd just like to acknowledge Jennifer Cavett, who's the collaborator on this, and we work together, both of our labs and a number of very really excellent PhD students who've carried out the work. Thank you.
Thank you for the excellent talk, Dennis. Next up, we have Xuefeng Chao from the Utrecht University.
Okay, so hi everyone. So the title of my presentation today is Campylobacter jejuni permeabilizes the host cell's membrane by shorting lysophosphatidylethanolamines. So bacterial membrane is composed of the phospholipids, glycolipids, and the proteins. Phospholipids are composed of the 2 fatty acids thiol and a variable head group, for instance, the PG and the PE. So lysophosphatidylethanolamines is a derivative of the phospholipids, which one of the acyl chain has been removed by phospholipase A, including lyso-PE, lyso-PG, lyso-PA, and the lyso-PS. Our previous study reported that there are 4 major phospholipids classes, including PG, PE, lyso-PE, and lyso-PG in Campylobacter jejuni. And in Campylobacter jejuni, membranes consist up to 45% lysophospholipids, including around 30% lyso-PE, 15% lyso-PG, and only very tiny amount of lyso-PA. No lyso-PC has been detected, while in other bacteria, the average lysophospholipids are less than 1%. This is indicated lysophospholipids is a very important molecule for Campylobacter jejuni. So here, based on the hemolysis experiments, we found the Campylobacter jejuni A116 wild-type bacteria cause the stronger hemolysis to horse erythrocyte compared to the phospholipase A mutant strain, indicating that Campylobacter phospholipase A is involved in bacterial hemolytic activities. Wild-type membrane isolation contains the same ability as the live bacteria, but the PODA mutant didn't, and protease K treatment didn't really affect the membrane isolation's hemolytic activity, revealing this compound might be membrane lysolipids. Further experiments were amplified by membrane lipids isolation for major phospholipids, lyso-PG, lyso-PE, PG, and PG were identified and purified. Hemolysis results demonstrated Campylobacter's hemolytic activity is achieved by lyso-PE. Additional experiment performed by commercial lysolipids found there is no significant differences in lyso-PG 18, PE 16, and the negative control D PPS. However, we found lyso-PEs are able to trigger hemolysis to erythrocyte. Moreover, with the decreases of the length of the acyl chain for lyso-PE, more toxic for erythrocyte has been found, indicating short chain lyso-PE is more harmful for erythrocyte. This observation is correlated with Campylobacter jejuni phospholipidome, which around 4% lyso-PE 14 and 3% lyso-PE 16 has been detected in Campylobacter jejuni membrane. Similar experiments were processed to detect whether short-chain lyso-PE is harmful for tissue culture cells, by measuring LDH released from cells. Two different cells types were performed here and revealed that phospholipase A also involved in casing leakage in epithelial cells. And the key factors in this process is also lyso-PE. And the length of the lyso-PE has been also shown affect its cytotoxicity, which the increase of the length of the fatty acid tile, less cytotoxicity has been observed, indicated short-chain lyso-PE14 is the most harmful for cells. So further study tried to explain how the lyso-PE induced the cell injury. Two kinds of the oxidative inhibitor, vitamin E and the DPPD, were used in this study. According to the results, that with the increase of the concentration of the oxidative inhibitors, less hemolysis observed, suggests that lyso-PE triggered oxidative events involved in lyso-PE induced cell injury. So to summarize, two key points in here. So Campylobacter jejuni lysophospholipids are toxic for erythrocyte as well as epithelial cells. Of the lysophospholipids of Campylobacter jejuni, the short-chain lyso-PE14 is the most harmful for cells. And the final, thanks for all the help from our group, and thanks for your listening.
Thank you, Xuefeng. That was very nice, and thank you for staying in time. If you get any questions, we'll come back to you at the end of the session. So now we have 3 or 4 short talks that are pre-records. So I will share my screen. Hopefully we can watch these smoothly. So the first one is Blaž from the University of Ljubljana.
Greetings, my name is Blaž and I'm a PhD student at Biotechnical faculty, and today I'll be presenting our work with the title Protein Profile of Campylobacter jejuni biofilm cells under temperature and oxygen conditions. One of the most important characteristics of this bacterium is its ability to survive in aerobic environments despite its microaerophilic nature. Biofilm formation, as shown in the picture, has been suggested to be one of the strategies used by this pathogen to persist in aerobic environment. The aim of this study was to evaluate proteomic profile of Campylobacter jejuni biofilm cells cultured at 42, 37, and 30 degrees at microaerophilic conditions and 30 degrees at aerobic conditions. Biofilm cells were grown for 48 hours on Miller-Hinton agar plates. After incubation, they were harvested and mechanically lysed. After lysis, proteins were purified and identified by labeled mass spectrometry. We grouped the most interesting detected proteins according to their function, as shown in this picture. It is important to notice that in aerobic mature biofilms, ferric uptake regulation and heat shock proteins were upregulated, while most of the flagellar proteins were not expressed except those associated with oxidative stress response, indicating that during oxidative stress, cell motility is reduced and the main focus of cells is survival. This approach allows us to study the environmental adaptations and resistance mechanisms of Campylobacter jejuni biofilm cells in relation to various stressors. It will also provide insights into the response of cells within the biofilm and reveal important molecular targets for limiting Campylobacter contamination and thus making food production safer. I would like to thank my mentor, Associate Professor Dr. Anja Klančnik, my former mentor, Professor Dr. Polona Jamnik, and our Associate Dr. Marjorie Fournier. The authors would also like to thank Slovenian Research Agency for funding the research project and also Project Instruct Ultra. And now I would like to thank you for your time and attention. If you have any further questions, please write me down to my email written below.
Okay, so that was a talk from Blaž, who I believe is here also today. So now we move on to the second presentation, which is from Heather Chick, University of Swansea.
Good morning all. Thank you for attending my talk today on pre-treatment of chicken epithelial cells with intracellular pattern recognition receptor agonists, leading to alterations in C. jejuni strain invasion and cytokine phenotype. I'm Heather Chick. I'm based at Swansea University and I work as a part of the BBSRC funded Campylobacter consortium. Extra-intestinal spread of C. jejuni in broiler chickens represents a significant public health threat, with some pathogenic strains possessing the ability to invade intestinal epithelial cells, migrate across the gut wall, and disseminate into edible tissues. There is good in vivo evidence in broiler chickens that C. jejuni-induced local and systemic inflammation may be responsible for compromising epithelial cell barrier integrity and may be a prerequisite for extra-intestinal spread. Understanding the specific stimuli that link these factors is integral in designing intervention methods for Campylobacter control. Using cell culture techniques, avian 8011 intestinal epithelial cells were treated for 6 to 24 hours with various pattern recognition receptor agonists to mimic an inflamed gut before addition of reference strain M1, in vivo high invasion LE55, and in vivo low invasion LE17 for 4 hours. Gentamicin protection assays revealed that pre-treatment with intracellular agonists poly-IC and MDP resulted in the low invasion LE17 invading significantly more into 8011 cells than both reference M1 and high invasion LE55. No differences in invasion were observed in human CACO2 cells, and despite distinct in vivo invasion differences, LE55 and LE17 showed similar invasion profiles in vitro. Simultaneously treating 8011 cells with transfected poly-IC and the same strains for 6 hours led to significant changes in cytokine expression profiles as determined by qRT-PCR. CXCLI1 transcripts were significantly increased in response to transfected poly-IC up to 4 log-fold, a relationship that was unaffected by the inclusion of C. jejuni despite a log-fold reduction. CXCLI2 transcripts in comparison significantly increased in response to the combination of transfected poly-IC and C. jejuni, but not in the individual treatments, with the exception of strain LE55, which induced low levels of CXCLI2 regardless of the inclusion of transfected poly-IC. These results represent differential regulation of inflammation during co-infection. And this data suggests a potential role for the intracellular toll-like receptor 3 and viral agonists in reducing the immunotolerance of chickens to C. jejuni, and also potentially represents a significant implication in the enhancement of C. jejuni-induced inflammation, and therefore the increased dissemination of C. jejuni into edible tissues. This is especially important when we consider that each treatment alone does not initiate a strong CXCLI2 response and is only enhanced when both are in combination. The high in vivo invasion strain LE55 both invaded less than the low in vivo invasion strain LE17 and did not initiate a strong CXCLI2 response when in combination with poly-IC, suggesting strong differences between these distinct C. jejuni strains. There is also a suggested role here for CXCLI1, long considered the non-functional homologue of human IL-8 in viral infections. Therefore, anti-inflammatory intervention strategies must focus on the type of inflammation generated, especially with regards to any co-infections that might be present within a flock and the differential immune regulations taking place. Thank you for listening.
Okay, so that was a really nice talk from Heather, and I believe she's here at the conference also. So the next one, the penultimate, is from Amy Hillier, also from the University of Swansea. So I shall load that up.
Hello all, I'm Amy, a PhD student at Swansea University funded by Chemin. Today I will present the potential of organic compounds to reduce the growth and extra-intestinal spread of Campylobacter within broiler chickens. Campylobacter poses a major public health threat as the primary cause of bacterial gastroenteritis. In 2019, there were 220,682 confirmed cases of campylobacteriosis in the EU, compared to 87,923 salmonellosis cases. Poultry is the main reservoir for Campylobacter and rapidly spreads through flocks horizontally via fecal shedding and coprophagic behaviour. The flock is processed and products end up on supermarket shelves where 80% are contaminated with Campylobacter. Consumption of undercooked poultry is the main entry route for the bacteria into the human GI tract. The bacteria make its way through the human digestive system, causing nausea, vomiting, abdominal pain, and fever. Most cases are self-resolving, but 10% result in hospital admission, and a small proportion of affected individuals develop sequelae. Throughout its journey from farm to fork, there is a risk for the bacteria to incur millions of dollars worth of costs due to productivity loss and illness. It is vital that the spread of Campylobacter be controlled at the source to minimize the economic and medical burden it poses. Campylobacter is a natural inhabitant of the poultry GI tract and is now accepted that there is a strain-dependent pathogenicity seen within chickens. When infected with a pathogenic strain, we see a dysregulated immune response induced by activation of toll-like receptor 4 and 21, which causes excessive inflammation and facilitates the extra-intestinal spread of Campylobacter. Inflammation alters the epithelial barrier, and major pro-inflammatory cytokines such as interferon gamma and TNF-alpha interfere with permeability. The solid black line on the image demonstrates the paracellular movement of Campylobacter due to compromised tight junctions, and the dashed line demonstrates Campylobacter's ability to move transcellularly. Many chickens appear asymptomatic with infection, but there are noticeable symptoms. Diarrhea is most frequently noted and leads to wet litter, which causes pododermatitis and hotmarks. On-farm biosecurity measures are not 100% effective at reducing Campylobacter, and there is no available vaccine. There is a huge gap in the market for an effective product that is economically viable, sourced from organic compounds, effective at reducing GI numbers of Campylobacter, and limits the extra-intestinal spread. In this study, we look at 3 potential compounds that can be utilized in this manner. Chromium propionate is a key organic mineral shown to affect cell function. The compound significantly increased metabolic output in our avian cell line at higher concentrations. We investigated any indirect or direct effects of chromium on Campylobacter invasion and growth using 5 isolates and found that chromium had no bactericidal effect on Campylobacter and no effect on inhibiting invasion. Caprylic acid has shown to be bactericidal against pathogens. We saw no cytotoxic effect of the caprylic acid on our cell lines, but there was a significant increase in human cell line metabolism. Isolates that were challenged with caprylic acid showed a significant decrease in growth at the 0.25 and 0.75% concentrations. When we pre-incubated cell lines with caprylic acid for 24 hours prior to Campylobacter challenge, we saw a dose-dependent decrease into our human cell line. However, this was not statistically significant. Butyric acid was chosen for its proposed bactericidal effect and role as an intestinal metabolite. No significant cytotoxicity was recorded, but spikes in metabolic outputs at higher concentrations in human cell lines was seen. When applied directly to Campylobacter isolates, we saw a dose-dependent decrease in growth across all 5 isolates on average, with the 2 highest doses being significant. Invasion of Campylobacter was decreased into the human cell line in a dose-dependent manner. The 2 short-chain fatty acids may have had an indirect effect on translocation of Campylobacter within the chicken by increasing gut health, and a direct bactericidal effect on Campylobacter, reducing load within the chicken gut. Chromium has shown to have no bactericidal effect, but may have an indirect effect on the gut health by affecting mitochondria within avian epithelial cells. Current work will be extended to include 4 probiotic options for feed supplements. With regards to the compounds presented here, we look at the inflammatory response elicited by our Campylobacter isolates and quantify this using cytokines. We will investigate the ability of the short-chain fatty acid and mineral compound to reduce this. The transepithelial electrical resistance of cell monolayers will be measured for untreated cell lines, Campylobacter challenge cell lines, compound challenge cell lines, and a combination. Campylobacter may be able to compromise tight junctions between epithelial cells, enabling them to pass more readily from the intestinal lumen into the bloodstream. We will aim to visualize tight junctions using microscopy and confirm presence using PCR to measure key proteins. To confirm the assumption that chromium is improving the metabolic activity of our avian cell lines, we will measure the oxygen consumption and quantify this from the mitochondria of our avian epithelial cell line. Thank you for listening to the presentation today, and here are the references that I have used.
Okay, so thank you to Amy from the University of Swansea, and we have the final pre-recorded presentation now from Alessandra Piquerillo. I hope I pronounced that right, from the University of Padua. I shall just load that up now.
Good morning, everybody. I'm Alessandra Piccirillo from the University of Padua, Italy. I'm going to present the virulome analysis of extra-intestinal Campylobacter jejuni and Campylobacter coli from commercial chickens. Campylobacter jejuni and Campylobacter coli are commonly known to colonize the ceca of domestic poultry without causing any sign of disease. However, in the last years, an increasing number of studies has shown that these two Campylobacter species have the potential to spread extra-intestinally. In the past, Campylobacter jejuni has been associated to a pathological condition named avian vibrio hepatitis and causing hepatic focal necrosis. Very recently, another pathological condition, spotty liver disease, has been associated to Campylobacter hepaticus. In this study, we present the results of a whole genome molecular characterization of 9 Campylobacter jejuni and 5 Campylobacter coli from livers or spleens of broilers, layers, and capons. A comparative genomic analysis was performed with the aim to identify common virulence factors associated with the extra-intestinal spread and possibly with the pathological lesions observed at necropsy and resembling those caused by avian vibrio hepatitis. The 14 isolates were fully sequenced by using Illumina technology, and raw sequences were de novo assembled by SPADES. Genomes were characterized by 7 loci and core genome multilocus sequence typing, whereas comparative genomics consist of whole genome-based phylogeny and virulome characterization. In this table, genome characteristics are summarized, but overall, a wide genetic diversity was detected among our isolates. Indeed, MLST showed 12 different STs, of which one was novel, while core genome MLST detected 13 different sequence type, and among the 14 strains, only 2 shared the same profile. This wide genetic diversity was confirmed by the core genome K-MER-based phylogenetic analysis, and the phylogenetic analysis highlighted that our isolates were scattered throughout the phylogenetic tree and clustered with isolate of poultry, turkey, and human origin. Hierarchical clustering analysis detected 133 virulence-associated genes involved in motility, adherence, invasion, and toxin production. A higher number of virulence-associated genes than the reference genomes was detected in our isolates, and when considering our isolates and the reference genomes of chicken liver or spleen origin, 12 common virulence-associated genes were detected. Finally, 7 virulence-associated genes were unique to our isolates. To conclude, the whole genome molecular characterization of Campylobacter jejuni and Campylobacter coli from livers and spleens showing pathological lesions resembling those observed in avian vibrio hepatitis suggests a possible association between them. The comparative virulome analysis revealed the common virulence factors between our isolates and reference strains, and a higher number of virulence factors in our strains. And this finding suggests a potential involvement of these factors in the extra-intestinal spread and possibly development of the disease. Finally, we suppose that environmental and/or host-associated predisposing factors such as immunosuppressive disease might contribute to extra-intestinal disseminations of Campylobacter jejuni and Campylobacter coli in the chicken body. Thank you for your attention.
Okay, so that concludes all the talks. Thank you very much to Alessandra Piccirillo from the University of Padua. I believe she's here also. So what I propose now is that all presenters, they probably should turn their cameras on because there will be questions coming their way. I see Arno has turned his camera on. Heather, check. Thank you very much for your presentations. And maybe we'll kick off. So Janie, maybe you can start with the first question. And I guess they could be in mixed order as well.
So I have a question here for Arno from Ben Pasco. Are surveillance programs unbiased?
It's a very good question. They're less biased than the way that people selectionize it because it's interesting, but they'll never be a completely unbiased one. There's always a reason why you do your surveillance. So whether it's food, animals, supermarkets, but even in the supermarkets, they won't swap everything. They will still swap the meat in this case. So there's already a bias towards meat. It is the best we can do. And it's much better than using just any isolate because somebody found it interesting.
Thanks, Arno. I have another question for you from Desi. I'm not aware of the USA's antibiotic law. Are there restrictions on these?
There is no law. There is a ambitious plan without any enforcement. So the answer is yes, they would like it to, but not if they, it's a bit almost like the vaccination discussion that there is. Yes, you should vaccinate, but we won't force you.
Great.
But they're trying.
Right. Ozan, would you like to ask a few questions?
Yeah, well, there's another one for Arno that I've got here. Well, maybe it's been answered. Comments to Arno. Not all aminoglycosides are equivalent. There is, for example, almost no resistance to gentamicin in Europe in Campylobacters. Arno, I'm not sure if you've already answered that.
I did answer that because the question came in direct, but there is a bit of gentamicin resistance in the data set, but it is indeed very low. Just because of time, I couldn't discuss every detail, but there are a few gentamicin UK resistant isolates, both C. coli and jejuni, and there are more gentamicin resistant isolates in the US, but it's still very low compared to the others.
Okay, thank you. I've got one more question here for Dennis, Dennis Linton from Arno. Iron is not usually an effluxed, rather expensive way of dealing with it, especially when there is a ferritin and a bacteria ferritin / DPS to store iron. Why would Campylobacter need that?
Yeah, I answered this, I think, to Arnie. It's a good question. Probably the iron exporter that we've characterized is more involved in trafficking of iron into the periplasm where it may be needed for different proteins. That's our best guess. It's probably different for the zinc exporter, which might be more direct zinc toxicity. We should also be aware that iron can accentuate the reactive oxygen species stress, so it may also be involved in responding to reactive oxygen. Okay.
Yeah, there's another question for Dennis Linton from Tom Wilkinson. Your macrophage system, survival system seemed interesting. Can you expand a little more on this? Are they human, chicken, primary activated, or what else?
So they're raw macrophages. So I've answered that in the discussion.
There's also another question for Dennis here from Chris Bayliss. How widespread amongst CJ strains are CZCD and CJ0948 strains?
As far as we know, certainly for CZCD, they're universal amongst C. jejuni strains. I don't think we've seen any strains that don't have CZCD. CJ0948, I'm pretty sure the same is true, but it's a bit more of a guess with that. So I don't think they're strain specific.
Thank you, Dennis. I've got a question here for Heather Chick from Tom Wilkinson. I'm not sure if it's been answered. As your supervisor tells me what we should do next, are there differences in intracellular and extracellular TLR signaling? That's for Heather.
Yeah, so my next step is actually to investigate what intracellular pathways are being activated upon inclusion in transfected poly-IC and C. jejuni, and in particular focus probably on toll-like receptor 4, which is extracellular, and toll-like receptor 3, which is intracellular and bound to an endosome. So I'm going to be doing this by including a variety of inhibitors to pre-incubate them before I then analyze my CXCL1 and CXCL2 production. And these are going to include more downstream signaling molecules such as NF-kappa B, perhaps some interferon-related factors, especially with relation to toll-like receptor 3, as well as looking at inhibiting toll-like receptor 3 and toll-like receptor 4 themselves to see if there is actually interaction at that initial stage. And then also to look at other factors such as MYD88, which is universal to the majority of chicken toll-like receptors, as well as looking at other intracellular agonists such as NOD2 and NLRP3 in the inflammasome.
Fantastic, thank you, Heather. I've got one more here. This one is for Amy from DESI. What are the benign and pathogenic strains? Also, can both benign and pathogenic strains be co-infecting the chickens? Very interesting talk, also commented.
So the benign and pathogenic strains, the collection of Campylobacter that I used, the isolates that I used were from free-range chickens. So we had some that had come from the ceca, the gut, and the liver. And when we've plotted these, we've noticed that the liver strains are slightly more invasive than the other two, where they've been isolated from. But there are still some, and when we say invasive, I mean they can kind of spread extra-intestinally. But when we've looked at the cecal and the gut strains as well, there are some that do stand out as slightly more invasive as well. And yeah, I mean, both benign and pathogenic can infect chickens, but it just depends on the, what we're going to do now is look at the inflammatory response that the benign and pathogenic strains induce. Well, what we believe are benign and pathogenic based on their invasivity, and see if this correlates as well. So we'll see if the ones that we believe are more pathogenic are actually inducing a higher inflammatory response as well in the avian cell line.
Thank you, Amy. Janie, Cynthia, if you'd like, if you see any more questions.
Xuefeng Chow, I have a question for you from Ozan. Does Campy, is that from you, Ozan?
Yes.
Does Campylobacter affect cellular lipid rafts, which ultimately could lead to cellular toxicity?
I'm sorry, can you repeat the question one more time?
Yeah, sure. So
Yeah.
does Campylobacter affect cellular lipid rafts, which could ultimately lead to cellular toxicity?
Yeah, that part we are not really investigating into that. We only through the lyso-PE, which react with the cells directly, and to measure the LDH which is released into the environment. So we believe that maybe lysophospholipase from the environment or from the Campylobacter itself, when they reach to the cells, they can cause some damage to the cell membrane. And through our oxidative inhibitor experiment, we believe that this process may involve some oxidative events in here. But actually, how exactly the lysophospholipase triggered the toxic for the cells, we still need to further investigate that.
Thank you.
Sorry, Cynthia, go ahead if you were going to say something.
There's some questions from Nabil Farid Alikham. He has one question for Dennis. How widespread among C. jejuni strains are the CZCD and CJ0948 strains? Is Dennis still there?
No, I think we answered that one. Sorry.
Was already answered? Okay,
Yeah.
sorry, I missed that.
And it wasn't me who asked it.
Okay. And then, yeah, go ahead.
Yeah, sorry, I was just going to say there's a question for Blaž. Blaž, the oxidative stress proteins in biofilm are down expressed or overexpressed. And I think Morgan Nenning has just posted one link to that saying, can you tell me, thanks for your talk, can you tell me which ones are overexpressed? Blaž, are you there?
Yes, I'm here. So thank you for your question. Yeah, regarding aerobic conditions in biofilms, there are all sets of overexpressed proteins, and most of them are trying to protect Campylobacter cells from oxidative oxygen molecules. So we detected, I don't know, chaperones, Gruel and Grues that are preventing from misfolding of proteins, DNA binding protein Hu that protects DNA from damages, and of course, I don't know, elongation factor 2 for production of new proteins. We also detected ferredoxin and I know DNAK that regulate oxygen molecules with iron ions. So I hope that answers the question. Thank you.
Thank you, Blaž. Any other questions coming through?
I have a
If
question
not,
here
yeah, we have one.
for
Excellent.
Xuefeng from Katie Davies. Have you looked at the lyso-PE content of C-jejuni OMVs?
Yeah, I assume this question is about if the other Campylobacter jejuni also contain such a high amount of lysophosphatidylethanolamines. Yeah, we did compare the different Campylobacter strain, especially Campylobacter jejuni. They are not contain exactly the same amount of lysophosphatidylethanolamines for sure. But the Campylobacter jejuni A3.6, which we used in this study, contains the highest lysophosphatidylethanolamines. We also tested the Campylobacter jejuni another Campylobacter jejuni strain. They roughly contain, if I remember correct, 30% of the lysophosphatidylethanolamines. So we believe that in Campylobacter jejuni, membrane lyso-PE is a key factor to cause the cytotoxicity. But for sure, different Campylobacter jejuni strains contain a different amount of lyso-PE. Yeah, I hope that is a correct answer. Thank you.
Thank you very much. Any further questions for our presenters or comments or anything? If not, then I would like to thank all the session presenters. Thank you very much. Fantastic talks to the co-chairs, Cynthia and Janie, and all of you for attending. The session will be continuing now in Discord. So please, as session 1 yesterday, do join in, talk to the presenters, discuss your topics, and that will continue until session 3 later on when Nicole Jenico will host session 3. Thank you very much, and I hope you enjoyed the session and see you all soon.